Hello, 9th standard Cbse children. Have a nice day. Have a good day. This is our video number 6. We are finishing this chapter, completing this chapter 5, Fundamental Unit of Life as part 6. Okay. As such, we have done in previous session, video 5, we have done cell division, and then part of video 4 as cell ordinals, and then we have discussed assignments. Is it not? But still, this cell division, I would like to have with the picture diagrams. I want you to understand still better. Hence this video, and this video will be for a short duration. Just understand the lesson. Okay. So this is video number 6, chapter 5, Fundamental Unit of Life. Last part of the lesson, cell division. So I'm completing this chapter as part 6. Okay. And as such, as I told you, the PDF that is sent to you, the lesson PDF that is sent to you may not have this cell division paragraph. That I have given it as a QB, page 65, 66. I given it as QB, page 65, 66. Textbook content, that is for this cell division. So you please understand that also. Okay. So now this cell division, as you know, we have, as I learned in a previous session, we have 3 types of cell division as amitosis, mitosis, and meiosis. Amitosis is a direct cell division, and mitosis is a indirect cell division, and meiosis is a reduction division. So I already told you. So this mitosis, it is taking place in the somatic cell or vegetative cell or in the body cells. Whereas amitosis, it occurs in the lower part, lower organisms, unicellular organisms, as amoeba, any other protozoans. It is amitosis, it is a irregular division. Otherwise, we say only the division of nucleus, it takes division of nucleus, and then there is a constriction appears in the cell wall. As the constriction deepens, the divided nucleus will get separated, and then 2 new daughter cells will be formed. That is for amitosis. Okay. Now this mitosis, it is a indirect cell division where the entire cell will take part. In that, there are 2 divisions. One is karyokinesis, first division of the nucleus, and cytokinesis, division of cytoplasm. Okay, so this is the introduction for mitosis, right? Now here, as I said, textbook page content 65, 69. Just follow it. Cell division, this is what given in our textbook. Sorry, as a 2021 edition, or I don't know which textbook we will get. I am not sure, but still I have given this. Right? So cell division, new cells are formed in organisms in order to grow, to replace old, dead, and injured cells, and to form gametes required for reproduction. Mitosis is taking place for this. The process by which new cells are made is called cell division. There are 2 main types of cell division, mitosis and meiosis. The process of cell division by which most of the cells divide for growth is called mitosis. I repeat, the process of cell division by which most of the cells divide for growth is called mitosis. And in this process, each cell called mother cell or parent cell that divides to form 2 identical daughter cells. The diagram given in our textbook. So that divides to form 2 identical daughter cells. I hope you understand this. The daughter cells have the same number of chromosomes as mother cell. What does it mean? For example, if human, there are 23 pairs of chromosomes. So as a result of mitosis, that new two daughter cells also will have the same number of chromosomes. So we say the chromosome number remains constant in mitosis, during mitosis. And it helps in growth and repair of tissues in organisms. So the last 2 points are significance of mitosis. Are it clear? So this is for mitosis. Then go through this for a second. Last 2 points. Daughter cells have the same number of chromosomes as mother cell. It helps in growth and repair of tissues in organisms. The next one, this is division of mitosis, cell division in during mitosis with the diagram. I am just going to explain. So it's not given in detail in our textbook. I want you to understand the lesson better. So now first division, it is interphase. As I said, mitosis, it is of 4 phases of nucleus, and first phase, the cell which is getting ready for mitosis, it is called interphase. Okay, so the interphase, before a cell undergoes mitotic division, it prepares itself for the division. This phase is called interphase. Now the chromatin material duplicates due to duplication of nucleic acid. Now at this stage only the nucleus is taking part. And as I said earlier, the division of nucleus, it is called karyokinesis. Then there are 4 phases for nuclear division as prophase, metaphase, anaphase, and telophase. I repeat, prophase, metaphase, anaphase, and telophase. I hope you follow. So this is the 4 phases in mitotic division. Now in prophase stage, chromatid fibers begin to coil and appears as long thread-like structures called chromosomes. I repeat, chromatid fibers begin to coil and appear as long thread-like structures called chromosomes. Okay, and each chromosome consists of 2 chromatids that lie side by side and are joined along a point called centromere. I repeat, each chromosome consists of 2 chromatids that lie side by side and are joined along a point called centromere. Right? Spindle fibers are developed from the poles towards the center, and nuclear membrane and nucleolus start disappearing. Now, see this diagram where this is a outer cell wall and then nucleus, right? Now the chromosome, can you see, they begin to separate. Here it is coiled, whereas here the first interphase diagram, it is coiled. Okay, whereas here it is getting separated, and this chromosome will undergo lengthwise splitting. So there are 4 structures peculiar, that is called 4 chromatids. They are known as chromatids. Then at the early prophase, we call it as early prophase, this uncoiling of the chromosome. The chromosomes become thicker, clear, and long thread-like structures. And each chromosome consists of 2 chromatids. Now let's the 4 chromatids, like the lengthwise splitting, and they lie side by side. First or a chromosome then at the end of prophase stage, the disappearance of nucleolus and nuclear membrane will happen. So I repeat, at the end of prophase stage, the nucleolus and nuclear membrane will disappear. அப்போ என்ன ஆகும்? Is a metaphase stage. Second stage is the metaphase stage where nuclear membrane totally disappears. Chromosomes become shorter and thicker. Chromatids move towards the center of the cell with their centromere and centromeres are attached to the spindle fibers. So I repeat the diagram for it. First diagram, chromosomes, they are moving to the center. So I say the chromatids are arranged on the equatorial plane and they are held by the spindle fibers. From where are they arranging spindle fibers? The spindle fibers, they are supposed to be from the centriole. During the time of cell division, spindle fibers will be arising from the centriole. I hope you could follow this. So, the nuclear membrane totally disappears. Chromosomes become shorter and thicker. The chromatids move to the center of the cell with their centromere. Centromeres are attached to the spindle fibers. This is for metaphase. So metaphase nale nee தூக்கத்துல கூட கேட்டா சொல்லணும். Chromosomes are arranged on the equatorial plane. Right? The next is anaphase stage, where the centromere of each chromosome divides into two. So here it is arranged. So now here it is dividing into two. And when each chromatid gets a centromere, it becomes a chromosome. நம்ம metacentric chromosome படிச்சோம்ல. Now, one of these chromosomes move towards one pole and other towards the opposite pole by the contraction of spindle fibers. So here, metaphase stage, they are arranged on the equatorial plane, and here the centromere divides into two and they are moving to the opposite pole as contraction of spindle fibers. This is for anaphase stage. Diagram go through panna. First is metaphase stage, second வந்து anaphase stage. நம்ம bookல இந்த diagram இல்லை, but I may ask you to draw this diagram. நான் QB ல தரல, but you have to follow this video and you have to draw the diagram. உடனே எதிர்த்து out of portion I didn't draw, she didn't give us. You have to follow this diagram. You start pass பண்ணி you can copy the diagram. You should have some extra effort. For that I am giving because the diagram is very simple. I hope you understand. Then next one, it is telophase stage. As we say it as telophase stage, early telophase and late telophase stage. So here the daughter chromosomes move to the each pole. The nucleolus and nuclear membrane reappear. Thus, two daughter nuclei are formed at the two poles of the cell. See here, the two chromosomes, at the end of anaphase stage, the chromosomes are moved to the opposite pole. Yeah? So this spindle fibers, they disappear and the reappearance of nucleolus and nuclear membrane will occur. And then in plant cell, there is a center cell wall formation that is called cell plate. There is a cell plate forming. I hope you understand. So till this early telophase stage, there is division of nucleus getting completed. And then division of cytoplasm. The division of cytoplasm is called cytokinesis. Okay? So this is early telophase, this one late telophase stage. Okay. So the daughter chromosomes reach the pole, the nucleolus and nuclear membrane reappear, and thus two daughter nuclei are formed at the two poles of the cell. And the spindle fibers disappear. This division of nucleus is thus called karyokinesis. Venn diagram பார்த்துட்டியா? So this is telophase stage. Next, cytokinesis. Division of cytoplasm. This is called cytokinesis. Okay. Now the division of cytoplasm is called cytokinesis. In plant cell, cytoplasmic division occurs by the formation of cell plate at the center of the cell between the two daughter nuclei. In our previous diagram, thus at the end of mitosis, two identical daughter cells are formed. This is the 4 stages of mitosis. What are all? Prophase, metaphase, anaphase, and telophase. Okay. Then significance of mitosis. This equational division. Mitosis is also known as equational division. Results in the production of diploid daughter cells, that is 2n, with the equal distribution of genetic material. Genetic material here it is DNA. Expansion of DNA, deoxyribonucleic acid. The multicellular organism growth, organ development, and increase in body size are accomplished through the process of mitosis. And mitosis helps in the repair of damaged and wounded tissues by renewal of the blastocysts. I hope you understand. Okay, so this is for the mitosis. And significance, you can learn these 3 points. Significance of mitosis, you please make a note and learn these 3 points. I hope you understand. Huh? Go through for 2 seconds. Mitosis, this is cytokinesis diagram. Again, I say it's not given in our textbook, but you have to follow this diagram. I may ask in the objective diagram போட கேட்பேன் இல்ல diagram கொடுத்து you may have to write what it is. Are it clear? So we can just pause this video and note down the diagram and significance of mitosis. Right? The next one, it is meiosis. Meiosis is the reduction division. Meiosis is the reduction division taking place in the reproductive cells. As I said the other day in previous videos, meiosis will occur only at the time of gamete formation. We say in female age attainment, otherwise puberty. That is the reproductive phase. Right? So right from the day 1, when the child is, the sex is determined as male or female, the development of sex organs, testis and ovary will start developing. Right? And then only when it is matured in female, when the ovary is fully developed, the release of ovum will occur. That is menstruation, or that is puberty, or that is age attainment. Right? Same way in male, there is a change in the body structure, change in the voice, and development of mustache and beard. Last year we have learned as reaching the adolescence, okay? And the characters will be formed. That is reaching puberty in both male and female. Okay, so that is the process of meiosis will occur. And in that, this meiosis again, there are 2 major divisions, as I said earlier in previous video, as meiosis 1 or heterotypic division and meiosis 2 or homotypic division. Meiosis 1 heterotypic division because it has again 4 stages as prophase 1, metaphase 1, anaphase 1 and telophase 1. In that, prophase 1 is the significant stages of division with the 5 substages. Those 5 substages are leptotene, zygotene, pachytene, diplotene and diakinesis. I repeat, as leptotene, zygotene, pachytene, diplotene, and diakinesis. In that, significant changes are taking place. As there is a variation begins here. We say during reproduction, the characters are passed on from one generation to the other through genes, is it not? And that is taking place here. Okay. So now in the leptotene state, you find chromosomes as blue and red part. Blue and red. One from father, one from mother. That we call it as paternal and maternal. Paternal father, maternal mother. Sperm ovum படிக்கிறோம் இல்லையா அதேதான். சரியா? In that, these two chromosomes, they are called homologous chromosomes. These two chromosomes are called homologous chromosomes. So during leptotene state, these chromosomes from the coiled position, they become uncoiled, clear and distinct. And zygotene stage, these chromosomes, they lie parallel to each other. They lie close and parallel to each other. And this is called pairing or synapses. Synaptonemal complex nucleus. So this is called pairing or synapses. This is taking place during zygote stage. So the characters of father and mother is getting attached. Okay. Then in pachytene stage, that is lengthwise splitting of chromosome, and they are called chromatid. And these chromatid, the diplotene stage, these chromatid, they into the chromatid stage, they become into like. And then they are attached at certain point. And that point of attachment is called chiasma. Chiasma singular, chiasmata plural. So at that point of attachment, crossing over will occur, and it is a tetrad formation. The tetrad formation, the chiasmata formation occurring. And when the chiasma occurs, there is interchange of segment, and that is called crossing over. I hope you understand. So here, for a chiasma, the blue, red attached circle, and attachment. So that is crossing. That phenomenon or that process is called crossing over. That will occur during pachytene stage. Okay. And then in diplotene stage, they become clear and distinct further. And in diakinesis, they move away from each other and they separate. That is called diakinesis. And that's the movement, the chiasmata, and the separate end they separate from each other. That is called terminalization. Diakinesis rendu chain pinni irukla adu but one stage they get separated. That is called terminalization. Then at the time of pachytene, diplotene and diakinesis, like that of mitosis, there is disappearance of nucleolus and nuclear membrane. And then with this exchanged segments, metaphase 1, anaphase 1 and telophase 1 will occur. So here in metaphase 1, the chromosomes are arranged on the equatorial plane and they are held by the spindle fibers. I hope you understand this. So this is for prophase 1 of meiosis and metaphase stage. The same meiosis in our textbook content, specific cell of reproductive organs or tissues in animals and plants divide to form gametes, which after fertilization give rise to offspring. And they divide by a different process called meiosis, which involves 2 consecutive divisions. And when a cell divides by meiosis, it produces 4 new cells instead of just 2. The new cells only have half the number of chromosomes than that of the mother cells. This is for meiosis. Okay. Then next one, adena meiosis எப்படி clear-ஆ கொடுத்திருப்பார். Peptide stage, nuclear membrane, and then this is condensation of chromosome இருக்கு. அதான் chromosomes become thicker and clear. And this is homologous chromosome. Blue and red and homologous chromosome. Correct? And then in zygote stage, I say pairing or synapsis, that we name it as formation of bivalent. As formation of pairing or synapsis as formation of bivalent. That is formation of synaptonymal complex. So we say synapsis between homologous chromosome. The pairing attached as a pairing. Correct? This homologous chromosome paternal and paternal chromosome when they come closer and like side by side. Then in the packaging stage, crossing over will occur. See here in the end segment, the single chromosome divides lengthwise splitting. So 4 chromatids are formed. And the 4 chromatids the blue and red are. So in the order blue, in the order red cross. This is homologous chromosome. Correct? So the inner one, it is an interchanged segment. Either in the blue, outer blue and red, they are free. Yeah? So the interchanged segments are the crossing over. Then, but there are many points of crossing over. So that region is called chiasma. Singular chiasma, plural chiasmata. And then at the diakinesis stage, the interchanged segment, they begin to move apart. That is called terminalization. The exchange segment blue, red, red, blue, and they are separated. So this phenomenon during diakinesis, it is called terminalization. I hope you are following this. Yeah? So this is for meiosis 1 as prophase stage. Okay. And then metaphase, anaphase, and telophase stage. In meiosis, it is like that of the mitosis only. But the exchange segments order, as I explained, it will be. I hope you understand. Yeah? So this is meiosis. And finally, as a result of meiosis, 4 daughter cells will be formed. During meiosis 1, prophase 1, metaphase 1, anaphase 1, and telophase 1, 2 daughter cells will be formed. And those 2 daughter cells will separately undergo mitosis. As a result, 4 daughter cells in the meiosis will be produced. Okay. The chromosome number, first it is 2n. When it comes to the second meiosis, it is reduced to half. That is n. 2n on the diploid length, haploid number of chromosomes will be formed during meiosis or reduction division. So meiosis helps to maintain the chromosome number constant in each plant and animal species. In meiosis, 4 haploid daughter cells are formed from a single diploid cell. This is very important in sexual reproduction during the formation of gametes. The occurrence of crossing over results in recombination of genes, and the recombination of genes results in genetic variation. And the genetic variations form raw materials for evolution. I hope you understand this. So this is for the cell division, mitosis and meiosis, right? I said in video number 5, I have explained But in the details, I am not actually that much fully satisfied for your explanation of video 5. So with this figure diagrams, I wanted you to understand still better. I hope you have done so. So this is for our video session number 6. With that, we are completing this chapter number 5, fundamental unit of life. Video file நம்ம வந்து என்ன சொல்லிருக்கேன், we have I have given the question bank and we have discussed assignments 1, 2, 3, 4 வரைக்கும் நம்ம முடிச்சிருக்கோம். So please follow that. Okay. So that runs for 1 hour, but still have patience and follow that. You would have done it. I hope you have understood. Yeah? So have a good day. Have a nice day. We'll continue in the next video session. Thank you.